Maturation of T lymphocyte. The maturation of lymphocyte from bone marrow stem cell consists of three types of processes: proliferation of immature cells, expression of antigen receptor genes, selection of lymphocytes that express useful antigen receptors. B lymphocyte mature in the bone marrow, T lymphocyte mature in the thymus. Each of the three processes plays a special role in the generation of the lymphocyte repertoire. The proliferation, here pre-antigen receptor expression, proliferation, antigen receptor expression, positive and negative selection. Here the failure to express pre-lymphocyte receptor, cell death occur. Here express one chain of antigen receptor. Here selection occur. Proliferation, immature B T cell express complete antigen receptor, strong antigen recognition. This is negative selection. Weak antigen recognition, mature T and B cell, positive selection. Interleukin 7 stimulate proliferation of B and T lymphocyte progenitor before they express antigen receptor, thus generating a large pool of cells in which diverse antigen receptor may be produced. Maturing lymphocytes are selected at several steps during their maturation in order to preserve the useful specificity. Selection is based on the expression of intact antigen receptor component and what they recognize. Cells that fails to express useful receptor die by apoptosis because they don't receive necessary survival signals. At the end of the process, the cell undergo positive and negative selection. Immature T cells are selected to recognize self-measure histocompatibility molecules. This process is called positive selection. After they mature, these T cells need to recognize the same measure histocompatibility molecule in order to be activated. The basis for positive selection is that antigen receptor on developing lymphocyte recognize measure histocompatibility molecule in the thymus and deliver signal for the survival and proliferation of these cells. Cells with correct self-measure histocompatibility restricted antigen receptor complete the maturation process. Immature B cell and T lymphocyte are also selected against high affinity recognition of self-antigen present in the bone marrow and thymus respectively. This process is called negative selection. Eliminate potentially dangerous lymphocytes that may be capable of reacting against self-antigen that are present throughout the steps in the maturation and selection of the measure histocompatibility. MHC restricted T lymphocyte, stem cell double negative, CD4 CD8, pre-T cell double positive CD4 CD8, strong recognition of the either class 1 class 2 measure histocompatibility. Apoptosis negative selection occur. Weak recognition of class 1 measure histocompatibility. Mature CD8 T cells positive selection. Weak recognition of class 2 measure histocompatibility peptides. Mature CD4 positive selection. CD4 T cell, CD8 T cell positive selection, and here the apoptosis negative selection. Production of diverse. In general, the germline organization of antigen receptor gene loci in the germline inherited antigen receptor gene loci contain coding segment exon that are separated by segments that are not expressed introns. HIG heavy chain constant C region or TCRC region consists of multiple exons that encode the domain of the C regions. L leader sequence, small stretch of nucleotide that guide protein through the endoplasmic reticulum and is cleaved from the mature protein. V variable, D diverse, here the H chain, extracellular domain, transmembrane and cytoplasmic domain, here loci, kappa chain loci, T cell receptor, beta chain locus, chromosome 7, and TCR and alpha chain locus chromosome 14. L leader sequence, a small stretch of nucleotide that guide proteins through the endoplasmic reticulum as they cleaved from the mature protein. The V variable, the D diversity, G joining, and C constant segment. Recombination and expression of Ig gene. The expression of Ig heavy chain involve two gene recognition event. DG joining, followed by joining of the region to the D complex with deletion and loss of intervening gene segments. The recombined gene is transcribed and the DVDG segment is spliced on the first heavy chain RNA, which is mu, to give rise to the mu messenger RNA. The messenger RNA is translated to produce the mu heavy chain protein. The recombination of other antigen receptor gene, the Ig light chain and the TCR alpha and beta chain followed essentially the same sequence, except that in loci lacking D segment, Ig light chain and TCR alpha AV chain recombine directly with a G gene segment. Here, the germline DNA, the recombined DNA into B cell clones, recombined DNA into B cell clones, primary RNA transcript, messenger RNA, Ig mu chain into B cell clones. Diversity in immunoglobulin Ig and the T cell receptor is produced by random combination of P, D, L, G gene segment, which is limited by the number of these segments and by removal and addition of nucleotide at the VG or VDG junction, which is almost unlimited. By animation you can understand this better. We'll show this in animation. Both mechanisms maximize diversity in the CDR3 regions of antigen receptor protein. Also, diversity is increased by the ability of different Ig heavy and light chain or different TCR and alpha and beta chain to associate in different cells, forming different receptors. Although the upper limit on the number of Ig TCR proteins that may be expressed in very large, it's estimated that each individual contains in the order of only 10 raised to power 7 clones of B cells and T cells with distinct specificity and receptors. In other words, only a fraction of the potential repertoire may actually be expressed. Number of V gene segment, immunoglobulin and T cell receptor. Number of diversity D gene segment, number of joining G gene segment, mechanisms, combinational diversity, number of possible VDG combination, junctional diversity, total potential repertoire with junctional diversity. Steps in the maturation and selection of B lymphocyte. The maturation of BC lymphocyte proceeds through sequential steps, each of which is characterized by particular changes in Ig gene expression and in the pattern of Ig protein expression and the pro-B cells and pre-B cell stages. Failure to express functional antigen receptor Ig heavy chain or Ig light chain respectively result in death of the cell by default pathway of apoptosis. Steps in maturation and selection of B lymphocytes. The IgG DNA RNA, DNA RNA, Ig DNA RNA, stem cells, pro-B, mature B, the germline D, stem cells none, Ig expression, germline DNA, here recombined H chain, gene, VDG, mu or messenger RNA, cytoplasmic mu and pre-B receptor associated mu, immature B, recombinant H chain, gene, kappa or lambda genes, and mu and kappa or lambda messenger RNA, membrane IgM. Mature B, alternative splicing of primary transcriptor to form C. Steps in the maturation and selection of major histocompetent restricted T lymphocyte. The maturation of T lymphocyte in the thymus proceed through sequential steps that are often defined by the expression of the CD4 and CD8 receptor. The TCR beta chain is first expressed at the double negative pre-T cells stage, and the complete TCR is expressed in the double positive cell maturation, culminate in the development of CD4, CD8 signal positive T cell, as in B cells. Failure to express antigen receptor at any stage lead to death of the cell by apoptosis. Steps in the maturation and selection of the major histocompatibility restricted T lymphocytes, base stem cell, and double negative CD4, CD8, pre-T cell, double positive CD4, CD8, the weak recognition, mature CD4, weak recognition, mature CD8, strong recognition of either class 1, class 2 apoptosis, and